Assalamu alaikum friends. Hope so that you all will be fine. Friends, in this video I am going to discuss about H.M.P shunt pathway. In this video I will discuss about the introduction and the pathway, the steps of H.M.P shunt pathway. And this will be my first part of this video, and in next part I will discuss about the significance of H.M.P shunt pathway and disorder. Okay friends, if you like this video then kindly subscribe to my channel. Okay, so let's start our lecture with the introduction of H.M.P shunt pathway. Okay, introduction. Friends, this H.M.P shunt pathway, it is an alternative pathway for the oxidation of glucose. Okay, now why this is called as shunt pathway? Because the some steps of glycolysis they are used in this pathway, so that's why it is called as shunt pathway. And in this pathway there is a synthesis of single phosphate, so that's why it is called as hexose monophosphate shunt pathway. Okay, there are some other names of HMP shunt pathway that are pentose phosphate pathway. Okay, so this is an alternative pathway. Alternative pathway for glucose oxidation. And why it is called as shunt pathway? Because some steps for glycolysis are used in this pathway, so that's why. And in this pathway there is synthesis of only single phosphate, so that's why it is called as HMP shunt pathway. And there are some other names of HMP shunt pathway, and that are okay, and that are first one is pentose phosphate pathway, because in this pathway there is synthesis of number of pentoses, so that's why it is called as pentose phosphate pathway. Pentose means 5 carbon atom containing sugar. The other name is, it is also called as phosphogluconate pathway, because this is the first intermediate that forms in this pathway, so that's why it is called as phosphogluconate pathway. The another name is Dicken, Dicken and Dicken and Horecker pathway. Dicken and Horecker pathway. So these are the other names of HMP shunt pathway. Okay, this is the introduction. These are the other names of HMP shunt pathway: pentose phosphate pathway, phosphogluconate pathway, and Dicken and Horecker pathway. Now coming toward the there are steps. Now coming toward the steps. Okay. Now coming toward the steps. There are two steps of HMP shunt pathway. There are two steps of HMP shunt pathway. The first one is oxidative step, and another one is non-oxidative step. Friends, the first step is oxidative step, another one is non-oxidative step. Why oxidative step? Because in this step there is oxidation of glucose, so that's why it is called as oxidative step. This oxidative step, it occurs in liver, adipose tissues, ovaries, lactating mammary glands, RBCs, testes, and ovaries. So oxidative step, it occurs in liver, adipose tissues, ovaries, lactating mammary glands, RBCs, testes, and ovaries. Okay. This oxidative step, it is very important for the production of NADPH. It is very important for the production of NADPH and is very important in the synthesis of fatty acid, steroid hormone, and cholesterol. Okay. The steroid hormones includes cortisol, aldosterone, and sex steroid. Sex steroids include estrogen, progesterone, and testosterone. Okay, so this oxidative step, it is very important in the synthesis of NADPH, which is very important in the synthesis of fatty acid, steroid hormone, and cholesterol. This non-oxidative step, it occurs in all cells. It occurs in all cells of human body and is very important in the synthesis of ribose 5-phosphate, which is very important in the synthesis of DNA, or we can say nucleotides, which is very important in the synthesis of nucleotide है काफी है nucleotides, and these nucleotides, they are very important in the synthesis of DNA. Okay, so these are two steps of H.M.P shunt pathway: oxidative step and non-oxidative step. This non-oxidative step occurs in all cells of human body, which is very important synthesis of 5 carbon atom containing sugar, that is ribose 5-phosphate, and it is very important in the synthesis of nucleotides. And nucleotides, they are very important in the synthesis of the DNA and RNA. Okay. Now, oxidative step, it occurs in the liver, adipose tissues, ovaries, lactating mammary glands, RBCs, testes, and ovaries. It is very important in the synthesis of NADPH, which is very important in the synthesis of fatty acids, steroid hormone, and cholesterol. Friends, now coming toward the pathway of the H.M.P shunt. Okay, pathway. Okay, pathway. So फोकस से आपने देखना है इस पाथवे को, थोड़ा कॉम्प्लेक्स है लेकिन आसान, इतना मुश्किल भी नहीं है. What happens that glucose 6 phosphate and the 6 molecule of glucose 6 phosphate, they gets entered in this pathway. How many molecules? 6 molecules. Okay. With the help of glucose 6 phosphate dehydrogenase enzyme, with the help of glucose 6 phosphate dehydrogenase enzyme, and there is a here NADP plus, it converts into NADPH and H plus, and we will consider the 6 molecules of here NADP plus, and same way here. Okay, the six molecules of glucose 6 phosphate with the help of glucose 6 phosphate dehydrogenase enzyme, and there's removal of 6 molecule of NAD plus which converts into 6 molecule of NADPH and 6 molecules of H plus, and there's a formation of phosphogluconolactone. Phosphogluconolactone, and there are also 6 molecules. Okay. Now in next reaction with the help of phosphogluconolactone hydrolase enzyme, sorry, gluconolactone hydrolase enzyme, with the help of gluconolactone hydrolase enzyme, there is a incorporation of water molecule H2O6 molecule. This converts into phosphogluconate. It converts into phosphogluconate. Okay, and there is also 6 molecules phosphogluconate. In next reaction, with the help of phosphogluconate dehydrogenase enzyme, okay, there is conversion of NADP plus into NADPH and H plus. 6 molecules here, there are 6 molecules, there are also 6 molecules. And also there is a loss of carbon dioxide. Okay. There is a formation of, okay, there is a formation of, sorry, there is a formation of ribulose 5 phosphate. There is a formation of ribulose 5 phosphate. There are also 6 molecules. Okay, ribulose 5 phosphate. Okay, so this is the oxidative step. This is the oxidative step. Here, this is the oxidative step. This is the oxidative step. This is the oxidative step. Friends, this is the regulatory enzyme in this pathway, glucose 6 phosphate dehydrogenase enzyme. And it is, this step is also rate limited step, or we can say it is a slowest step in this pathway. So this is the oxidative step. There is a formation of ribulose 5 phosphate 6 molecule. Now in next reaction there is the action of two enzyme. There is action of two enzyme, and that are epimerase, there, and that are epimerase and isomerase. And the 6 molecules of ribose 5 phosphate, it converts into 4 molecules of xylulose 5 phosphate and 2 molecules of ribose 5 phosphate and 2 molecules of, okay, and 2 molecules of ribose 5 phosphate. Now there is the, अब यहाँ से ribulose 5 phosphate से नॉन ऑक्सीडेटिव स्टेप स्टार्ट हो चुका है. There is action of two important enzyme epimerase and isomerase enzyme. It leads to the formation of 4 molecules of xylulose 5 phosphate and 2 molecules of ribose 5 phosphate. Now मैं इसको इरेज कर रहा हूँ, यहाँ से हम दोबारा से स्टार्ट करेंगे. ओके, क्योंकि जगह नहीं है इस वजह से यहाँ से हम स्टार्ट करेंगे. ओके, मैं यहाँ से स्टार्ट कर लेता हूँ. Okay. Four molecules of there are four molecules of xylulose 5-phosphate and two molecules of ribose 5-phosphate. जगह की तंगी की वजह से, क्योंकि आगे इस पाथ स्टार्ट ही मैंने यहाँ पर लिख लिया है. Now here there is action of transketolase enzyme. The action of transketolase enzyme. Transketolase enzyme. There is action of, मैं उसको दोबारा से लिख लेता हूँ. There is action of transketolase enzyme. Okay. And this transketolase enzyme, for its activation, it requires thymine pyrophosphate. Okay. There is action of transketolase enzyme. And listen very carefully. Focus कीजिएगा यहाँ पर आप. It is a 5 carbon atom containing sugar, and there are 4 molecules. And we will consider in this reaction only 2 molecules. As these are 4 molecules, we will consider only 2 molecules. We will consider only these 2 molecules of xylulose 5-phosphate. 2 molecules we will use in later. Okay. Now, with the help of this enzyme, there is a transfer of 2 carbon from this keto sugar to this aldo sugar. Okay. This is a 5 carbon atom containing compound. 5 carbon, there is transfer of 2 carbon. 5 minus 2, that will be 3 carbon. And here there is 2 molecules, they get transferred from keto sugar to aldo sugar. And 5, okay, 5 plus 2, that is equal to 7. And there is a formation of pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. These are also 2 molecules. Okay. Listen, I am again repeating that with the help of this enzyme, there is a transfer of 2 carbon from this keto sugar to this aldo sugar, and 5 minus 2, that is equal to 3. That sugar will be glyceraldehyde 3-phosphate, and 5 plus 2, 7. That is the pseudoheptulose 7-phosphate. 2 molecules on each side. Now, next reaction, there is action of transaldolase enzyme. There is action of transaldolase enzyme. Okay. Transaldolase enzyme. With the help of this enzyme, there is a transfer of 3 carbon from this keto sugar to this aldo sugar. 3 carbon. Okay. This is the 7 carbon atom containing compound. 7 minus 3, that will be equal to 4. And here 3 plus 3, that is equal to 6. And there is a formation of erythrose 4-phosphate and fructose 6-phosphate, and these are also 2 molecules. Okay. there is a transfer of 3 carbon from this keto sugar to this aldo sugar, and this is 7 carbon atom containing sugar. 7 minus 3, that is equal to 4, and 3 plus 3 equals 6, and there is a formation of 6 carbon atom containing, that is fructose 6 phosphate, and 4 carbon atom containing sugar, that is erythrose 4 phosphate, 2 molecules on each side. Now we will use these 2 molecules of cellulose 5 phosphate. Okay, now we will use 2 molecules of cellulose 5 phosphate. मैं इसको erase कर रहा हूँ. Okay, now we will use the 2 molecules of xylulose 5 phosphate. Xylulose 5 phosphate, 2 molecules. Okay, this is now. Now there is action of again transketolase enzyme. There is action of again. There is action of again transketolase enzyme. Sorry. Transketolase enzyme. There is action of transketolase enzyme, and it requires thymine pyrophosphate for its activation. With the help of this enzyme, again there is a transfer of 2 carbon from keto sugar to aldo sugar, and this is a keto sugar, and here 5 minus 2, there is 3 carbon, and this is a 4 carbon atom containing sugar. 4 plus 2, that is equal to 6, and it leads to the formation of fructose 6 phosphate and glyceraldehyde 3 phosphate. These are the 2 molecules. These are the 2 molecules, and these are 2 molecules. Okay, 3 carbon atom containing sugar, glyceraldehyde, it is a 3 carbon atom containing sugar, and this is a 6 carbon atom containing sugar. Now the 2 molecules of glyceraldehyde 3 phosphate, through reverse of glycolysis process, it form only 1 molecule of the fructose 6 phosphate. Through reversal of glycolysis, it forms only 1 molecule of fructose 6 phosphate. Only 1 molecule of fructose 6 phosphate. Okay. Now, in this pathway, in this step, there is a generation of 5 molecules of fructose 6 phosphate. 2 molecules are here, and 2 molecules of fructose 6 phosphate here, and 1 molecule here. Totally, there is a synthesis of 5 molecule of fructose 6 phosphate. Okay, 5 molecule of fructose 6 phosphate, and these 5 molecule of fructose 6 phosphate, with the help of this 5 molecule of the fructose 6 phosphate, with the help of reversal of glycolysis process, through reversal of glycolysis process, they form 5 molecule of glucose 6 phosphate. They form 5 molecule of glucose 6 phosphate. Okay friends, this is the non-oxidative step. This is the non-oxidative step. This is only remember the function of this enzyme, transketolase enzyme and transaldolase enzyme. Okay, it is very little bit complicated, and must remember. This is the tricky point. This is with the help of this enzyme, there's transfer of 2 carbon from keto sugar to this aldo sugar, and 5 minus 2 3, that is 5 plus 2 7. This is aldo sugar, it is a keto sugar, and there's a formation of pseudoheptulose 7 phosphate and glycerate 3 phosphate, 2 molecules. And in this case, we are considering 2 molecules. Now, this action of transaldolase enzyme, and with the help of this enzyme, there is a transfer of only 3 carbon from keto sugar to aldo sugar, and 7 minus 3, that is equal to 4, and 3 plus 3, that is equal to 6. It is a keto sugar, it is a aldo sugar. And there is a formation of ractose 4 phosphate and fructose 6 phosphate, 2 molecules on each side. Now we will use these two molecules of xylulose 5-phosphate, and again there is action of transketolase enzyme, and in this enzyme it is responsible for the transfer of 2 carbon from this keto sugar to this aldo sugar, and 5 minus 2 is equal to 3, and 4 plus 2, that is equal to 6. Okay, and there is a formation of glycerol 3 phosphate and fructose 6 phosphate, 2 molecules on each side, and 2 molecules of glycerol 3 phosphate through reversal of glycolysis process they form 1 molecule of fructose 6 phosphate. Now in this pathway, in this step, there is a formation of 5 molecule of fructose 6 phosphate, 2 here, 1 here, and 2 here. And through reversal of glycolysis process, this 5 molecule converted into the 5 molecule of glucose 6 phosphate. Now I'm writing the overall reaction. The overall reaction is 6 molecule of glucose 6 phosphate, they gets entered in this pathway, and there's also 12 molecules of NADP plus they gets incorporated, and 6 molecule of H2O, and ultimately there's a formation of 5 molecule of glucose 6 phosphate and 12 molecule of NADPH, and 12 molecule of H plus and 6 molecule of carbon dioxide. So this is the overall reaction. Okay, so this was all about the H.M.P shunt pathway. Okay, keep watching, watch again and again, and this way you can easily memorize, and you can easily write these steps of H.M.P shunt pathway. Keep watching, and thank you.